A very good morning, my dear students. We are here to discuss about unit number 2. I would like to give a brief introduction about unit 2, which is cell cycle and its regulation. In this chapter, we are going to learn about how cell divides, what are the factors responsible for the division of a cell, and how cell division is going to be controlled by different factors. And later on in the last part of this chapter, we are also going to learn some genes which cause cancers. So let's get started with this chapter. So as we know, cell division is a very essential event for survival of living organism, whether it is unicellular or it is multicellular. Cell division, especially mitotic division, is essential. So as we can discuss this by taking an example. When we took birth, we were of only 3 kgs or 2.5 kgs or 2 kgs. But as we started growing up, now we have gained a weight of more than 50 kg, more than 40 kg, whatever it is. So this gain in weight or this increase in the size of an organism is due to the division of cells, mitotic division of cells. So there are 2 things which are essential in respect of cell division. If the cell divides in a normal way, the organism will be leading its normal life. If cell division stops, ultimately it leads to death of an organism. Or if cell division is in excess, rapid cell division is there, it may lead to some diseased conditions like cancer. So cell division should go in a normal way. So it should not stop also, it should not overexpress also, over cell division or lot of cell division. If it is occurring inside the body of an organism, it may lead to the cancerous conditions. And as we know, mitotic cell division includes the different phases, like there are 4 main phases of cell cycle. The first phase is called G1 phase. The second is S phase, and the third one is G2 phase. So the G1, S and G2 all together, they are considered as interphase. So they are recognized as interphase. So this interphase is a stage where cell prepares itself to undergo cell division, and the remaining phase in which nuclear as well as cytoplasmic division occurs is termed as M phase. M is for mitotic phase. So this is a phase in which you are going to get mitotic division of nucleus as well as cytoplasm. And as we have studied this mitosis in previous classes, we know that there are 4 subphases of M phase. One is prophase, second is metaphase, third is anaphase, and fourth is telophase. So there are 4 sub phases of M phase, and in case of interphase you are going to get 3 phases G1, S and G2. So this is a preparatory phase of a cell for undergoing cell division, and M phase is a phase where actually there is a division of nucleus as well as cytoplasm takes place. So as of now, what we have understood is interphase is of very little importance and M phase is very important. So that's why we have studied various phases of M like prophase, metaphase, anaphase and telophase in detail in our lower classes, but we have not concentrated on interphase like G1, S and G2. But at PG level, we are going to concentrate more on these interface stages and less on M phase. So it takes, while I will tell you, it takes nearly 23 hours to complete the interface, and it takes only 1 hour in case of mammalian cells. M phase is completed in only 1 hour, whereas 23 hours are required for interphase. So which is most important, which is the stage which is taking longer time is most important. So interphase is the most important phase of cell cycle, and that's why we are going to concentrate on this interphase. So in the G1 phase, whenever the cell gets a signal to divide or whenever there is a need of cell division, the cell will enter into G1 phase with by receiving some signals, external signals or internal signals that you should divide. Then only it enters into G1 phase. G1 is actually preparatory phase for undergoing S phase. So as we know, in S phase, the DNA content or chromosomal content of the cell will be duplicated. So this S stands for synthetic phase. In S phase, there is a replication of DNA to make 2 copies to be distributed in the daughter cells. So whatever is necessary for DNA replication will be synthesized in the G1 phase. Then the cell, once everything is ready, everything which is necessary for undergoing replication is ready, then the cell enters into S phase and it undergoes DNA replication, and then after completion of DNA replication, the cell will enter into G2 phase. So this G2 phase is a preparatory phase to undergo M phase. So the proteins and other necessary things, necessary materials required in the M phase are synthesized in the G2 phase. That's why it is called as gap 2. So in some cells, one more additional phase of interphase, subphase of interphase you are going to get, which is G0 phase. G0 phase. So when the cell feels that the material required for undergoing DNA replication is not ready, the cell will undergo into G0 phase. Instead of entering into S phase, it enters into G0 phase because there is no need of division, or the materials for actual division are not yet synthesized inside the cell. When it feels, it enters into G0 phase. Like there are some examples of human cells which permanently enter into G0 phase, and the examples are nerve cells and muscle cells. Many times we have heard that if there is an injury to the nerve cell, it cannot be repaired or it cannot be replaced, because the nerve cells are permanently entering into the G0 phase. They will never come back to the S phase. Hence, cell division in case of nerve cells is very, very difficult as they are found permanently in the G0 phase. So nerve cells cannot divide. In some cells, for time being, the cell will enter into G0, and whenever everything is ready, then again from G0, the cell will enter into S phase. There are some cells which temporarily enter into G0, and there are some cells which permanently enter into G0, and one such example is nerve cell and muscle cell, which is found permanently in the G0 phase and it has lost its cell division activity. Then in case of S, as we know, DNA replication occurs, but who will give the signal for entering into the S cell? So you should get some signal that everything is ready and every raw material required for synthesis is ready, then you should start a synthesis. So that signal is provided in the late G1 phase, late G1 here. Here the cell will get some signal to enter into the S phase, and this signal is called as G1S signal. Then one more signal we are going to get is here G2M phase. G2M phase. So when cell undergoes DNA replication, and whenever every other material required for undergoing M phase is ready, the cell will get a signal that you should enter into M phase. Everything is ready now, you are allowed to enter into M phase. During that time, the cell will enter into M phase. If it won't get the signal here in the late G2, it will not enter into the M phase. So why this gap 2 is created? Because one thing is to produce the materials required for M phase, and another thing is to check the mistakes done in DNA replication. If there is a replication error, or if any mistake has been done during the DNA replication, it has to be corrected in the G2 phase. So it takes longer time to correct it, and then the cell is allowed to enter into the M phase. Otherwise, it will not be allowed to enter into the M phase. So this is first checkpoint. This is second checkpoint, and this is third checkpoint. So there are 3 checkpoints in the cell cycle. One is at G1 S phase entry, other is G2 M phase entry, and third one is at metaphase to anaphase entry. So in this phase, the checkpoint will check whether all the spindle fibers have attached to all the chromatids, chromosomes or not. If they are attached, the cell is permitted to undergo anaphase, enter into the anaphase. If spindle fibers are not attached to all the chromatids of a chromosome, then it is not allowed to enter into the anaphase. So spindle fiber attachment to the chromosomes is necessary to separate out the chromosomes. If they are not properly attached to the kinetochore region of a chromosome, then equal division of chromosomes will not result. Hence, it is necessary to stop or pause the cell cycle in this phase, metaphase to anaphase entry. The checkpoint will check whether spindle fibers are attached to the kinetochore or not. If they are attached, they will be allowed to enter into the anaphase for proper segregation of chromosomes, otherwise it won't. So first, second and third. At these three points, the cellular machinery will check whether everything is okay to enter into the next phase. It's like a closed door. It will always be closed. So whenever you show your identity that I am ready to enter inside the room, then only you are permitted to enter into the next room. So here once again your identity will be checked, and again here also one more time it will be checked for different processes. So at three different points we have checkpoints. So these are called checkpoints. there are three checkpoints. One is G1S, second is G2 to M, and third is metaphase to anaphase checkpoint. So we'll stop our discussion here and we'll continue with the positive regulation and negative regulation of cell cycle in the